Assalamualaikum warahmatullahi wabarakatuh, and a very good evening to my fellow friends and Madam Zulinda. So my name is Nur Nabilah binti Ramli, and today I'm going to present my first slide, which is the genetic engineering. So basically, what is genetic engineering? Alright, genetic engineering refers to the direct manipulation of DNA to alter an organism characteristic, which is the phenotypes, in a particular way. Genetic engineering, also known as a genetic modification or GM, which the process of altering the DNA in an organism genome. This may mean by changing or deleting a whole region of DNA, or even introducing an additional copy of a gene. It may also mean extracting DNA from another organism genome and combining it with the DNA of that individual. So genetic engineering is used by scientists to enhance or to modify the characteristic of an individual organism. For example, genetic engineering can be used to produce plants that have a higher nutritional value or can tolerate exposure to herbicides. So, moving on to the next slide, which is who is the founder of genetic engineering? Alright, genetic engineering was first found and used by a biochemist that is known as Stanley and Herbert. They were an American biochemists who were among the first to cut the DNA into fragments, and then they rejoined different fragments and insert the new genes into an E. coli bacteria, which then reproduce. So they both found or start using genetic engineering in the year of 1973. Right. By mean that when a genetic engineering is used, there must be a lots of application of genetic engineering. For example, it is used in medical, where through the recombinant DNA techniques, bacteria have been created are capable to synthesize human insulin, human growth hormone, alpha interferon, a hepatitis B vaccine, and other medically useful substances. While in agriculture, genetics engineering and biotechnology is used to help modifying plants, animals, and also microorganisms to improve the agricultural productivity. The application in agriculture can be modifying the genotypes of crop plants to make them become more nutritious, productive, and able to resist disease. While application of genetic engineering in disease is that genetic disease can be possibly corrected by replacing the dysfunctional genes with normal functioning genes. So basically, genetic engineering genetic engineering aims to modify the genes to enhance the capabilities and gives better health for organism. So moving on to the next slide, which is the example of genetic engineering. Firstly, we have bananas. In many countries around the world, bananas are considered as the main source of calories. According to reports from Uganda, their production is compromised by the emergence of new disease. Ugandan scientists have successfully used a genetic modification by inserting a paper gene into bananas, which prevent the fruits from getting the disease. Secondly, examples of genetic engineering that has been achieved is onion that don't cause watery eyes. The introduction of a single gene that decreases the function of the onion enzyme that makes water for your eyes has succeeded. And lastly is carrot that helps to prevent osteoporosis. An American research team has transferred a gene from the mouse ear cress into the carrot so that they contain more calcium. So in 2018, A test was conducted to mice and 30 volunteers, and it shows that humans absorb about 45% of the calcium compared to the original carrot. So, for the advantages of genetic engineering is it improves in pharmaceutical works. It creates a better food product where genetic engineering makes it possible for us to manufacture food products with a better nutritional profile. It means that from less food items or small portion of food, we can get what we need nutritionally. In addition, more food can be sent to parts of the globe where food poverty is a big issue. So basically, we all not only get to eat healthy foods, but when they are correctly engineered, more people get to benefit from nutritionally rich foods. Besides, genetic engineering can also increase the rate of maturity that can be achieved for products within our food chain. And it can also be disease resistant, produce medicines or medical treatment that will help billions of human, and lastly it could stop genetic disease in human. So lastly, for the genetic engineering disadvantages is that it is a technology that can be easily abused, where we fear that they could contribute to the introduction of unwanted and potentially harmful traits into microorganisms that were previously free from them. For instance, is an antibiotic resistant toxin development or a likelihood to trigger disease. Secondly, is the changes level of diversity species that we have. It seems like genetic modification or GM will enhance diversity, but it actually reduces it. This is because when a favorite product performs well, it becomes the industry priority. Number three is that it has the possibility to create another disease in organism. And lastly, it is not just DNA sequence that can be patented genetically. It may also be possible to patent seeds and crops. For farmers who live near fields where transgenic crops have been grown, and technically the transgenic crops has caused some problem where the genetically engineered crop seed has been distributed to other area and triggering accidental development of the genetically modified seeds to the land where they fall.
Now, genetic engineering is one of those topics that can seem
Please
quite
watch
complicated at
this
first,
video before you
but it's
proceed
really
to the
quite
next
simple.
slide. Thank
The
you.
basic idea is that we find one organism that has a desirable characteristic, like a plant that has lots of big fruit. Then we take the gene that's responsible for that trait and transfer it to another organism, so that this other organism develops the same trait. So effectively, what we're doing is modifying the organism's genome by adding a new helpful gene. And so we can call the organism genetically modified, or GM. We're not just limited to organisms of the same species, like we've shown here though. We can also use the genes of one species to genetically modify organisms of another species. We've now genetically engineered all sorts of genes into all sorts of organisms. For example, some sheep have been genetically engineered so that they can produce substances like drugs in their milk, which we can then extract and use to treat diseases. In a similar way, we've genetically engineered bacteria to produce the human hormone insulin, so that we can harvest it and use it to treat diabetes. And we've genetically modified crops to do all sorts of things, from improving the size and quality of their fruit, to becoming resistant to diseases, insects, and herbicides. Scientists are now researching how we could use this idea to treat some inherited disorders that are due to faulty genes. The idea, known as gene therapy, works by giving a person the healthy version of the gene in the hope that it will fix the problem. But it's proving pretty difficult to actually get it to work. One of the problems is that the faulty gene would be in all of the person's cells. So to fix this, we'd have to transfer the new gene into every cell in the body, which is really difficult. A potential solution to this, though, is transferring the gene at an early stage of development, such as at the egg or embryo stage. Because then, as the person develops, the healthy gene would get passed on to all of the other cells. As with everything in science, there are some potential problems with genetic engineering. So to explore these, let's take a quick look at the pros and cons of genetically modified crops. The pros are that we can easily make crops have desirable characteristics, like more edible fruit or being resistant to disease, which means we can make more food for less money. And this is particularly important when it comes to developing countries, as people still die from starvation. And we can also make sure that plants produce special nutrients. For example, golden rice contains beta-carotene, which can protect people from going blind. One of the main issues, though, is that we don't know for certain how genetically modified plants might affect our health. There's currently no evidence that GM crops are bad for us, but we just can't say with 100% certainty yet. There's also the chance that plants could make their way into the wild, where they might outcompete local plants and change the whole ecosystem. This concern is pretty unlikely, though, as the plants have been specifically modified to survive in a farmer's field, not to survive in the wild. The last thing we need to cover is how we actually transfer the gene from one organism to another. The first step is to find the gene that we want and then cut that section of DNA out to isolate it, which we do using enzymes. Next, we insert the gene into a vector, which could either be a virus or a bacterial plasmid, which remember are those little loops of DNA that bacteria have. We then introduce the vector to whichever organism we want to have the gene, for example a pig or a plant. And the idea is that the organism's cells will take up the vector and the useful gene it contains, and so they'll start producing the protein that the gene codes for. Anyway, that's the end of this video, so we hope you enjoyed it, and we'll see you next time.
Assalamualaikum warahmatullahi wabarakatuh. My name is Misa binti Muhammad Yunus and I would like to present about biotechnology. Biotechnology is a branch of applied science that uses living organisms most of the time in order to produce certain products. It is created and invented to give benefit for human economy, for instance in healthcare, agriculture, and environment. We will now move to the background of biotechnology. A common misconception is that we thought biotechnology is relatively new because it is only involved working with DNA and genetic engineering, which is not true. Biotechnology is not new. Humans have been manipulating living things to solve problems and improve their way of life for millennia. The origin of biotechnology dates back nearly 10,000 years ago when people collected seeds of plants with the most desirable traits for planting the next year. The term of biotechnology was coined in 1919 by Karl Erecki, a Hungarian engineer. At that time, the term meant all lines of work by which products are produced from raw material with the aid of living organisms. Between 1930 and 1952, researchers focused their effort on the relationship of genes and protein. They found a direct relationship between mutation of gene and the sequence of amino acids in a protein. Penicillin was discovered during World War II, which led to the focus of pharmaceuticals. The Cold War years were dominated by work with microorganisms in preparation of possible biological warfare and the production of more antibiotics. The final threshold to modern molecular biology was crossed in 1953 with the discovery of the structure of DNA by James Watson and Francis Crick. It was experiments by many other scientists soon followed to be determine how the information in the gene is decoded and expressed. Biotechnology brought industry and agriculture together at the beginning of 20th century. Fermentation processes were developed that produce acetone from starch and paint solvent to be used on automobiles.
We will now move on to the type of biotechnology. There are 4 types of biotechnology, which are medical, agriculture, industrial, and environmental. Under medical biotechnology, they have vaccines and antibody, while for agriculture, they have pest-resistant crops and plant and animal breeding. For industrial, they have biocatalysts, fermentation, and microorganisms. Lastly, for environmental, they have bioremediation. I will now explain about the type of biotechnology in details. First, medical biotechnology is the use of living cell and other cell material to better the health of human. Mainly, it is used for finding cure as well as getting rid and preventing disease. Vaccines are chemicals that stimulates the body's immune system to better fight the pathogen when they attack the body. They achieve this by inserting the weakened version of the disease into the body's bloodstream. Antibodies is tried that have been made in the development of antibiotics that combat many plants are grown and genetically engineered to produce the antibodies. Second, agriculture biotechnology focuses on developing genetically modified plants to increase crop yields or introduce characteristics to those plants that provide them with an advantage growing in regions that have some kind of stress factor on the plant, namely weather and pests. Biotechnology has provided technique for the creation of crops and express anti-pest characteristics naturally, making them very resistant to pests as opposed to having to keep dusting them and spraying them with pesticide. For the example, it will be when fungus genes being transferred to the crops. Selective breeding has been a practice. Humans have engaged it since farming began. The practice involved choosing the animal with the most desirable characteristic to breed with others so that the resulting offspring will have also expressed this trait. Third, industrial biotechnology is the application of biotechnology for industrial purpose that also include industrial fermentation. Applying the technique of modern molecular biology, it improves efficiency and reduces the multifaceted environmental impacts of industrial processes. Biocatalysts have been developed by the industrial biotechnology companies such as enzyme to synthesize chemical. Enzymes are protein produced by all organisms. The desired enzyme can be manufactured and commercialized quantities using biotechnology. While for fermentation, the corn sugar can be fermented to acid, which can then be used as intermediate to produce other chemical feedstock for various products. Some plants, such as corn, can be used in of petroleum to produce chemical. Microorganisms find their use in chemical production for the design and manufacture of new plastics or textiles and development of new sustainable energy sources such as biofuels. Last, environmental biotechnology is technology used in waste treatment and pollution preventing that can more efficiently clean up many waste compared to conventional method and significantly reduce our dependence on method for land-based disposal. Bioremediation refers to the application of biotechnical method which help the developing enzyme bioreactors that will not only pre-treat some industrial and food waste components but also allow their efficient removal via sewage system without using solid waste disposal mechanism.
Now we will look at the application of biotechnology. There are a lot of applications in biotechnology, but we will only focus on 6 of them. The first one is biofuels. Biofuels is one of the biggest applications of biotechnology, which in the energy production sector. With fears over the dwindling oil resources in the world, and their related environmental impacts, there is a growing need to protect the globe's future by finding alternative, environmentally friendly fuel sources. The second one is healthcare. Biotechnology is applied in the healthcare sector in the development of pharmaceuticals that have proven problematic to produce through other conventional because of purity concern. The third one is food processing. The method of fermentation using the microbial organism and their derivative is applied by which raw material that are inedible and easily spoiled are converted to edible and potable foods and beverage, which will have a longer shelf life. The fourth one is industrial biotechnology. Industrial biotechnology is the application of biotechnology that range from the production of cellular structure to the production of biotechnology elements for numerous sources. Next is wound dressing. Biotechnology also applied to the use of wound dressing coated with chitosan, which is a sugar that typically obtained from shrimp and crab shell. Last but not least is fuel from waste. Applying bioremediation waste can be converted to biofuels to run generators. Microbes can be induced to produce enzyme required to turn plant and vegetable material into building blocks for biodegradable plastic. We will now move to the traditional biotechnology versus the modern biotechnology. Let's focus on the traditional biotechnology first. In early biotechnology, it is includes of recombinant DNA technique, tissue culture, and mutagenesis. Next, breeding animals and crops to make cheese, yogurt, bread, beer, and wine. Microorganisms are used to produce various products such as enzymes for the use in laundry detergent. Last is involve using natural organisms to create or modify food or other useful products for human use. Now let's see what is the difference in the modern biotechnology. So, in modern biotechnology, they include DNA profiling, genome analysis, transgenesis, DNA cloning, and also tissue engineering. Modern technology techniques are used in many areas such as food, agriculture, forestry, healthcare, environment, minerals, and industrial processes. It also involves manipulation of genes and living tissue in the control environment. So as you can see, there are not much different, but there is a difference between traditional and modern. As you can see, the modern will have more area of using biotechnology compared to the traditional one. We will now move on to the benefits of biotechnology. The first one is improve our health. Biotechnology can help us to improve our health because it helps to improve the nutritional content of our food supply. Necessary vitamin and mineral can be produced in croplands, and this reduce health issues that are related to the lack of nutrients. At the same time, biotechnology also improve cropland yields and nutritional density, so people can eat less and still receive the same amount of nutritional values. That allows more people to have the food they need. Second is allow us to preserve resources. Biotechnology gives us an opportunity to extend the lifespan of our food. This practice includes salting our food to preserve them, freezing and drying food, also the method that we use to preserve our food that has been known for centuries. Heating food products to remove harmful elements also can be used to preserve for an extended period. The third one is reduce infection disease rate. Biotechnology has helped us to create vaccine. It has helped us to be able to create treatment that reduce difficult symptoms of disease. It has even help us to learn how infectious diseases can be transmitted so their transmission can be reduced. That allow us to protect those who at the most risk to the diseases and giving them a chance to live a happy life. Last is offer medical advance opportunities. Biotechnology allow us to look within just as easily as we look to the outside of the world for advancement. Studies that involve of human genome have allowed us to understand more about genetic diseases and some cancer, creating more effective treatment for them, and sometimes cures. It has allowed us to explore the reason behind certain birth defects to understand the importance of folic acid that makes it possible to extend average human lifespan. Last but not least, we will look into the risks of biotechnology. There are four major risks in biotechnology, which are health, environment, biodiversity, and socioeconomic. In health, potential health risks of genetically improved organisms relate to assessing and minimizing the risk of food allergen in genetically improved food. That an introduced protein in the food is not concerned if there is no history of common allergenicity, no similar amino acid sequence to nonce the allergen, rapid digestion of the protein, and also the protein is expressed at the low level, which is why it can risk our health. Next, environment. The risk of policymakers and regulators need to assess include the potential for the spread of threats from genetically improved plants to the same or related species plant. Biodiversity risks associated with the possible creation of new selection pressure coming from the introduction of genetically improved organisms into the environment. Lastly, for socioeconomic is when modern science may bypass the needs of poor people. Before
What
we
is
move on
biotechnology?
to
Since
the next
bio is usually
subtopic,
related to
please
living
watch this
organisms
video.
and
Thank you.
technology related to mechanics and computing, so perhaps biotechnology is related to bionic augmented superhumans? Actually, not quite so. Technology is the use of techniques and science to make products or modify processes. Biotechnology is the use of living organisms, aka bio, to make products or modify processes. Before we can go on to application, we have to understand how living things work, right? Indeed it is. Hence, every major breakthrough in biology understanding is followed by novel biotechnology. For example, in 1865, Mendel discovered the law of inheritance, which basically says that biological traits are passed on to the next generation through particular ratios and expressed according to dominance. This would later lay the foundation of artificial breeding in agriculture, where animals and plants are bred selectively to develop preferred biological traits. In modern day, the central principle behind biology lies in genetics, or DNA. DNA's building blocks, A, T, G, C, can be seen as sort of a life code that is universal to all living things. A string of these life codes, put together in a particular sequence, instructs specific protein formation, resulting in life as we know it. This is kind of similar to how alphabets in English can be regarded as communication codes and universal to all English words. A string of alphabets or communication codes put together in a particular sequence instructs a particular word, resulting in the communication as we know it. The technological application through the understanding of genetics resulted in many applications, ranging from GMO crops in agriculture to gene therapy in medicine. It usually employs the method of introducing DNA from one species to another, or silencing existing genes to achieve specific biological traits. Think about it in a certain way. It is kind of similar to forming new words like chillax, from chill and relax. So, in summary, biotechnology is the technological application of biology, oriented to serve our life. What are some biotechnology you could think of? And what is the future of biotech? Share it with us. What is biotechnology? Since bio is usually related to living organisms and technology related to mechanics and computing, so perhaps biotechnology is related to bionic augmented superhumans? Actually, not quite so. Technology is the use of techniques and science to make products or modify processes. Biotechnology is the use of living organisms, aka bio, to make products or modify processes. Before we can go on to application, we have to understand how living things work, right? Indeed it is. Hence, every major breakthrough in biology understanding is followed by novel biotechnology. For example, in 1865, Mendel discovered the law of inheritance, which basically says that biological traits are passed on to the next generation through particular ratios and expressed according to dominance. This would later lay the foundation of artificial breeding in agriculture, where animals and plants are bred selectively to develop preferred biological traits. In modern day, the central principle behind biology lies in genetics, or DNA. DNA's building blocks, A, T, G, C, can be seen as sort of a life code that is universal to all living things. A string of these life codes, put together in a particular sequence, instructs specific protein formation, resulting in life as we know it. This is kind of similar to how alphabets in English can be regarded as communication codes and universal to all English words. A string of alphabets or communication codes put together in a particular sequence instructs a particular word, resulting in the communication as we know it. The technological application to the understanding of genetics resulted in many applications, ranging from GMO crops in agriculture to gene therapy in medicine. It usually employs the method of introducing DNA from one species to another, or silencing existing genes to achieve specific biological traits. Think about it in a certain way. It is kind of similar to forming new words like chillax, from chill and relax. So, in summary, biotechnology is the technological application of biology, oriented to serve our life. What are some biotechnology you could think of, and what is the future of biotech? Share it to us.